Welcome, everyone. We're very excited to have you here today to talk about how tissue matters. My name is Jenna Lilly, and I'm the Executive Director of the Children's Brain Tumor Network. And I wanted to introduce Jen Mason. Jen, did you want to take it from here?
Yeah, so I'm Jen Mason, and I'm Director of Operations of the CBTN, and I'm going to go ahead and get started. And then we'll introduce our scientists later on when we're going to go into the questions section. So it's today's topics, if you want to go to the next slide. We're going to be talking about CBTN. So I'm just going to give a brief overview. I know a majority of people on the call already know what CBTN is about, but I'll do a quick overview. And then we're going to talk about tissue matters, why we collect tissue, and then what makes up tissue, and then ultimately how it will affect precision medicine. And then we have a few researchers on the call who we'll open it up to questions for them. Okay, next slide. And of course, we are going to be recording this so that we can post it later. So if you don't want to have your video or audio recorded, please put a message in the chat. And then of course, you're welcome to submit questions throughout the presentation, and then we can address those at the end. Okay, next slide. Okay, so let's talk about what the Children's Brain Tumor Network is and where we are today. So next slide. The challenge of a pediatric brain tumor is it's the leading cause of disease-related death in children, not just within the US, but across the world. And there's never been a drug developed specifically for pediatric brain tumors. So a colleague of mine actually likened herself to a mad scientist years ago, where she's trying to put together decades-old drugs to help treat her patients without the downstream toxic effects of those therapies. So unfortunately, she's working with something truly old. So we're trying to develop new treatments for these children. Next slide. So in comes the Children's Brain Tumor Network, which is formerly known as the Children's Brain Tumor Tissue Consortium. It's a network made up of researchers, clinicians, and of course our patients and foundations from around the world in order to help our children and to make sure they don't die or suffer from brain tumors and of course the downstream effects of those therapeutics. It's built on some core values, one being open science. We want to make sure that all the resources that we've put together, from the biospecimens to the clinical data, the path and op reports, the imaging, and of course any omic data that's generated, is open and available for anyone to access. And then of course a global inclusion is twofold. It's not just our members. We have institutions that are part of the consortium that are in Australia and Zurich, China, and Italy. So we're across the globe, but researchers across the world can access the resources that we've built. You don't have to be a member to access them. And we've had over 200 projects that have used our specimens, our clinical data, our omic data, and of course, they're not all members of the institutions, which I will go into a little bit later. And of course, pioneering spirit is another core value. CBTN was a novel idea when it first started. Institutions were actually a little unwilling to join CHOP to do this. They were saying, why would we send our specimens to you? But we have become trailblazers, for lack of a better way of saying it. And that also goes into the pioneering spirit of the young researchers that have an idea. So you don't have to have 20 publications and NIH grants. If you just have this great idea, you can access the specimens data that's available in the resource in the bank. So you just will support anyone with that pioneering spirit. Next slide. And this graphic actually talks about that traditional research model versus what CBTN does. The traditional research model, we're changing that. It's typically siloed. So a researcher in one lab may be, they try to collect tissue locally from the patients that are enrolling at their own institution, but it could take them years decades to collect as much as they possibly need to do their research, but also might be limited in scope to a specific diagnosis. Whereas CBTN, we're actually closing in on 4,000 at this point. I think we're a little over 3,800 enrolled. So all of those specimens, the tens of thousands of specimens, are available for researchers across the world to access. And if you think about it, one patient's worth of donation may actually end up supporting dozens, if not more, researchers from the tissue all the way down to any data that gets generated from that one particular sample. So again, changing the traditional research model and the traditional research landscape. Next slide. So let's go into Tissue Matters and why CBTN is collecting tissue and why it's so important to do so and to continually collect it until we help our children. So brain tumors are super complicated, as pretty much I think everyone on this call knows. There are over 125 different types of brain tumors, with new subtypes being discovered daily. So traditionally, a child might be diagnosed with, let's say, tumor type A. But now we know that with not just the histologic diagnosis, we're looking at the molecular underpinnings of that tumor. So they might be diagnosed now with tumor A with molecular features of tumor type B. And this is going to be key with respect to their therapy, because if two children are diagnosed with one's tumor type A, one's tumor type A with molecular features of tumor type B, they may react completely different to the same therapy. So it's important to know what those molecular underpinnings and the mutations that are going on within the brain, within the tumor, to find better therapies. And of course, these brain tumors are a depletable resource. So we are going to continually collect them, because the more we collect, the more we can understand. And of course, since there's over 125 different tumor types, we could never collect enough. Especially with the rarest of the rare, for example, ETMR, embryonal tumor with multi-layered rosettes. This typically affects children under 2, and really these children they're given a very poor prognosis, but they're very, very rare. So if we can come together and collect those, a researcher may only have 5 or access to 5 of those over the course of years. Whereas if we pull those resources together with all of the consortium members, we might have 50 and be able to generate a nice size data set for research. So the progress in finding the therapies for our children is definitely going to be significantly higher the more brain tumor tissue we collect and all the other resources. Next slide. And I'm going to actually pass this slide over to Jenna.
Sure, thanks, Jen. So I just wanted to take a second to talk about what Jen was saying about the complexity of the tumors. As Jen said, we know the tumors are very complex, but there are 10 common characteristics of cancer tissue that were identified by Dr. Weinberg and Hahnemann in 2000. And that was really a coalescence moment for researchers to understand the diversity and the complexity of research that was needed to really make progress in cancer research. And so we have an example here where CBTN research uses that little card on your right-hand side. We have a researcher that is actually looking at one of those 10 common characteristics in pediatric brain tumors. And so through leveraging this infrastructure of common characteristics, we can make sure that CBTN is making progress along those known mechanisms of cancer. The example that you're given here is that one of the mechanisms is called sustaining signaling, and that sustaining signaling is actually what tells the body to keep letting the tumor grow. Normally, your body has these brakes that it puts on when it identifies that something should be turned off. But this cancer mechanism actually pushes the gas and removes the brake. And so by looking at that specific signaling pathway that's going on in the cell, there's then an opportunity to get a drug that would put a brake back. And so through that oversimplification of this cancer mechanism, you could understand that we would not be able to study this cancer mechanism without actually having that tumor tissue and the data that goes along with it. I'll kick it back to you, Jen.
Okay. All right. So I wanted to talk a little bit about, what makes CBTN even more unique, aside from open access and sharing with anyone who has that idea. We are collecting tumors over time. So we aren't just collecting from one surgery. We are collecting from multiple surgeries. So if a patient has an initial CNS tumor, an initial tumor, and then we collect that tissue from surgery. Then we're following them in clinic to see how they're doing. Their tumor, we follow, or we capture what treatment they're receiving. And then if they have a recurrence or progression, we're capturing that tumor all again. And then, of course, setting the clock over. So we're going to follow them in clinic 6 months, 12 months, 18, 24. And then if they have another recurrence or progression, we're going to collect that tumor again. So what this tells us is, of course, the biology of that tumor, but also what's happening to that tumor over time with that treatment. So we know that some patients that are diagnosed with one tumor type, they're actually dying from another. So we're able to understand what the biological underpinning of what's going on with that tumor over time. So, and it's going to help identify the best treatments, again, going back to that molecular subtype. So it's very important that we collect those longitudinal samples along with the clinical data and really comprehensively put together a tremendous amount of resources for our researchers to help our children. Next slide. Now, this is going to go into what we can do with the tissue itself. So obviously collecting and generating the omic data is very important, but this is very important as well. So I'm going to do my best to describe cell lines and organoids, but we have some experts on the line that are going to be able to explain this further in a little bit. So cell lines are actually tumor avatars. They're a copy of the tumor that are grown outside of the body in dishes, and it's breaking it down into the cellular level. And cell lines are actually very challenging to grow, and they will grow based on the tumor type. So some tumors grow very aggressively into cell lines, whereas others are a bit more challenging. But they are a renewable resource. So we don't think of those as the tissue that we are getting from surgery, because they can be propagated again and again. So they're very valuable. They're like data, where they're not necessarily consumptive. We're not going to deplete them. So they're important to research. And the next slide are organoids, which can be generated much like cell lines, but they're 3D tumor avatars, are almost like mini organs that are generated outside of the body, and multiple things that can be done with these. Again, they're renewable. And this is something that Mateusz, who is on the call, that can talk about. We just started this process over the past year to develop these organoids. Of course, they can be banked for researchers to request downstream, targeted drug testing, and then using mouse models and injecting them into the mice, and then of course possible CAR T therapy. So there's a lot of things that can be done with these organoids, and of course Mateusz and the group will talk about these in a little bit. So tissue is very important, and the way we capture the tissue from surgery allows us to grow the cell lines and the organoids. Especially at CHOP, we are not only collecting the tissue in what's called freezing media to support the integrity of the tissue, but we're also getting fresh tissue from surgery as well. Next slide. And again, just as it's a renewable source of the biological sample, just restating that. It takes a lot of time and effort to grow these cell lines and organoids. It's not happening overnight. It can take weeks, months, or even up to a year to grow some of these, especially if they are typically tumor types that are challenging to grow the cell lines from. Next slide. And I think this is for you, Jenna.
Thanks, Jen. And I think you covered this a little bit, but the preclinical models are the cell lines and organoids and that Jen mentioned are critical to be able to test new drugs, to be able to look at the mechanisms that the cancer has, because you actually have the specific patient's tumor cells. And if you can grow them outside, then you have this model that you can test on that could potentially accelerate drugs where we would not want to give an untested drug to a child. So there's the ability to do large scale drug testing. We have projects where we take all of the FDA approved drugs and test them against our cell lines to see if there's targets. We also make sure that in the future, our hope is to be able to have these very patient-specific avatars that allow us to do real-time drug testing for that patient. And so these resources are a huge need for the research community. There are limited pediatric brain tumor models out there due to the nature of the disease, but also to the resources available. So CBTN has currently about 70 cell lines, and as Jen mentioned, it's just started the organoid work, but this is something that we believe that we can help empower the research community with. Can I get back to you, Jen?
And part of what can be requested are these models that are being generated. So right now we have, it's beyond 70 at this point, scientific projects that have been requesting using our samples in the biorepository. And as you can see from this graphic, it's not just the CBTN researchers that are requesting access to these resources. So again, it's inclusion of any researcher, not just within the consortium. Okay, so the next one is charting the unknown, or what makes up the tissue. And I'm going to actually pass this back to Jenna for the next slide.
So we talked about, as Jen said, this title of Charting the Unknown, and many of you have previously heard us say data generation. And you know, what does data generation actually mean? And what we're hoping to convey here is that without mapping what actually is the pieces of the tumor cell, the researchers cannot then study the mechanisms of what's going on in the cell. And so we currently right now have prioritized some data types. One you'll hear is called whole genomic data. And this actually shows us every single base pair that is present in the tumor's genome. And this allows us to see, this is an oversimplification that I'm saying, but some areas where maybe there is a nucleotide missing, or maybe it's in the wrong place. And if there is an abnormality within that genome, that gives researchers an understanding of how the tumor potentially starts, grows, or survives. We also look at RNA-seq data, and this actually tells us what genes are being expressed, and that is the next level in our cellular mechanisms. We have the genome, and the genes are then expressed. And at times we can see in cancer that some genes are overexpressed, underexpressed, altered, or completely missing. And that again gives researchers an idea of what's going on with that particular cancer that could potentially lead to a therapeutic. The other area we're looking at now is genomic data, proteomic data, and proteins are actually what is produced by the genes and the gene expression. And so with the proteomic data, we again look to see if there's an overabundance, the distribution or alterations of those proteins. This is another clue that would allow researchers to identify therapeutics. That sustaining signaling that I mentioned before would require whole genome data, RNA-seq, and proteomics to really give a comprehensive overview to a researcher of what's happening and how that brake was removed and how the gas pedal is on. Thanks, Jen.
Okay. And the next, so going with that genomic data, like how it helps us to understand pediatric cancer. So the genomic data can tell us why the tumor why the tumor started growing, if there are certain treatments that could be targeted based on that specific mutation. If there isn't a known treatment for a mutation, we might have one with another cancer. So for example, if there's a great treatment for uterine cancer with the same mutation as a medulloblastoma, maybe there's a possibility to use that therapy for the medullo. And then, of course, genomic information can help discover molecular reasons why so many rare diseases are connected. We know that children that are born with a craniofacial abnormality are more likely to have a brain tumor later in life. So the genomic information is very powerful, but it's all coming from that super informative piece of tissue. And then anything that we discover from that genomic information can be brought back to the bench and the laboratory for further discovery. Okay, and then of course, as I mentioned earlier, we have well over 70 scientific projects using our specimens and the cell lines, but now we also have over 120, and I know a few more came in just this past day, requesting access to the data that we've generated. So the omic data, the genomic information, whole genome sequencing, RNA sequencing goes into what's called the Pediatric Brain Tumor Atlas, or the PBTA, and that's available on a number of platforms. So Cavatica is actually our pediatric genomic cloud. So that's where all of the raw sequencing data sits. And researchers and bioinformaticians, and if you just have knowledge of genomics, you can go in there and use pipelines that are already in Cavatica and to run analysis on that raw sequencing data. And then there's, of course, the Gabriella Miller Kids First Data Resource Center. It's based on a grant that was NIH funded. And it's essentially a biorepository of, it's a file repository of rare diseases, so that researchers can come and look at craniofacial data and files that are available versus brain tumors. And then the pediatric PEDC Bio portal, my apologies, is a portal that has already analyzed data from, what essentially started out as raw sequencing data. So all of these platforms are available for researchers to access and see the data that we've generated from that very precious tissue. And again, there's a graphic, that has the non CBTN researchers. We have 77 versus 52. So more researchers that aren't part of the consortium have generated or requested access to the data than our actual members. So that's great. That's who we want to reach and access. Okay, thank you. Now, precision medicine. So this is what Jenna was talking about, the molecular underpinnings and being able to identify a targeted treatment for a patient based on what their biological makeup of their tumor is. So all I see is the title slide, so I don't know if you can advance it or, okay, there we go. Right now, we actually have a study currently underway where we are utilizing that genomic information and panel data that is being generated from these tumors that are being taken at surgery. It's being sequenced, and then there's a tumor board that's actually looking at all that data together to identify personalized therapy for that patient based on that molecular subtyping. So, and then of course, any data that's generated from that one patient is going right back into that pool of data to further inform on future patients. So again, like CBTN, we just want to keep the data coming back in to further the research and increase the number of resources and help future patients. So with that, we are just about 30 minutes in. I think the next thing is to introduce our scientists on the call. So if we want to go to the next slide, we have Mateusz Kaptier as the lab director of the preclinical lab within the center. Jolyn Rakita is our supervisory bioinformatics scientist within the bioinformatics unit. And then Pyle Jane is a research scientist also within the preclinical lab. So I will go ahead and open it up to questions. I'm not sure if anyone's posted questions in the chat because I was unable to keep up with that. But I guess, I don't know if Mateusz, Jolyn, and Pyle, if you want to chime in and provide any additional information on what Jenna and I presented, and then we can open it up to questions.
So as Jen introduced me, so I'm one of the research scientists who works in the lab. Some of my major projects really are focused on looking into what drives or what's the mechanism behind some of these really rare as well as some of the most common brain tumors in kids. And I really cannot also emphasize how important some of these models have been because they feed into my work from two different ways. Like one is from like the data that is generated, as Jolyn mentioned, that we can use iteratively in multiple different projects and ask multiple different questions and really try to hone in on the mechanisms that are causing these cancers in many different ways. And the second way all this contribution really helps drive my work is by the models and the avatars that we are talking about, because without the avatars, we really wouldn't know how the cancer will react to a specific drug that we will give the patient. So, so my work really focuses on these two avenues and really bring it together in a manner that we can that we work with clinicians in clinical trials and really bring back what we are finding in the lab to help patient on a very real-time basis. So I think that's also one other aspect of CBTN that I would like to highlight, that things happen pretty much in real time, and that's very fantastic, and it's just very impressive to see, because I remember as a young graduate student, I used to see or I would hear about these extremely rare brain tumor subtypes, and when I would start looking into what's known about them, there'd be like one paper or one case study. And now I think thanks to CBTN and other similar organizations that are all working together, there are so many models and so much like genomic data, which has really enhanced our knowledge about these tumors. And definitely like the next step and the ongoing step is to better treat these cancers. So thank you.
Thanks, Kyle. That was great. I think at this point it is an opportunity to ask any questions about CBTN, about tissue. We have our scientists here, and so they can answer specific questions that you have or just overview.
I have a question and also a statement. First of all, thank you very much for making this presentation tonight, for taking the important time that you have in providing this information, and of course for doing all this great work. My question, maybe this is more to Mateusz, because it seems to be the focus of his work, trying to link this up with the precision medicine. It looks like there's a timeline mismatch, that it takes an extended period of time and lots of work today to make these cell lines have you, what are your thoughts in trying to change that situation so that this data, the cell lines, can be more quickly available?
Thank you, Jim, for that question. That's actually a very, very important question, and I think that's a question which we deal basically every time, every day, I would say, with as new requests are coming for cell lines, and we would love to provide 25, not 5 cell lines for high-grade glioma at that point, or we would love to provide not 1 cell line of very rare tumor, but ideally 100. And it is actually a statement also to the fact that we really need to have access to more tissues and be able to develop more models to be stored and then using high throughput studies, which would to some extent replace this need of quick evaluation, because it is a power in there is a power in analyzing multiple samples. And we know that from adult cancers, that the fact that availability of for example, breast cancer, that access to cell lines and tissues for breast cancer in the past accelerated significantly the drug development. So that's one of the aspects which just drives our work every day, that we want to make sure that we can get those as soon as possible available for researchers. One thing which we cannot change, we cannot really, we could, but we don't want to really force these cells to grow faster. We could use artificial methods to make them so-called immortalized and grow very fast, but they will not fully represent then the ideal primary tumor model. And it's enough that we already putting them outside of the body and use them on plastic, making them slightly different, I guess, or at least pre-selected, having only part of the tumor really growing there. But since this is very important topic, and I want to just make this maybe a bit shorter, but there's for example, mentioned organoid system, which we see actually extremely important model for future development, because organoid as something different than cell line, organoid actually are a chunk of tumor, which will grow outside on plastic. So it's not really the so-called dissociation and making it a single cell level, but it's a combined normal stromal tumor composition, which still grows in plastic, but it won't grow forever. So once that grows, I think we see that's what we're right now evaluating, and see a very quick opportunity to use that in potential drug studies or any precision medicine based explorations within maybe not 4-6 months, but actually within a month or 2 for patients for which we could have those organoids in culture. And I think an entire field is really looking into this type of approaches as one of the opportunity. I know that tissue itself, there are actually attempts where tissue itself as a small chunk tissue is cultured for a relatively short time and explored as a quick precision medicine approach to evaluate a drug response within that space. But cell lines still as a not depletable resource, as building up those cohorts, which will allow us to introduce them constantly into new high throughput studies for potential targetable, druggable options, which then we can bring back to the precision medicine field and explore with each patient using either organoids or chunk culture as well, if that helps. Thank you.
I think just to add on to that, because I think a part of the question, Jim, was that we're a little concerned about the timeline of using those cell lines for precision medicine. But one thing that I want to bring up again, because Jen talked about it a little bit, was this clinical trial effort that we have going on in the center, which actually uses the whole tissue, excuse me. So it's a part of the PNOC Pediatric Preclinical Oncology Consortium. It's a high-grade glioma trial that we get whole genome or whole exome plus RNA sequencing data, and then we analyze and turn around that data within a 15-day time period. We review the findings and present at tumor board. So we do use the primary tissue from the brain in those cases and try to do a rapid turnaround for precision medicine efforts. And since we can't really do that rapidly with the cell lines at this moment.
Got it. Thank you.
That's a really great point. Any other questions? That was a great question. Thanks so much, Jim, for asking that. No, I'm very surprised. I think we have some, I think even maybe Kristen Gillette's on this call, and she normally has a question.
No fair on calling me out. I do not have a question, but thank you for asking.
Sure. Okay. So we must have been very clear. I hope we were very clear. Well, you know, I think one of the last slides that we had did provide ways to get in touch with us. And so we're always here to be able to answer your questions. We're so thankful for the participation of the patients and families. And we hope that you connect with us, ask questions. We will be sending a survey out that hopefully will give us some information on if this was helpful, did it meet your needs. We're doing these monthly meetings to help you be informed of what CBTN is, what are some of the challenges in the pediatric brain tumor research community, how we're trying to overcome them. And we look forward to topics that you want to hear more about. Okay, I guess with that, we'll say thank you so much for taking time to meet with us today, and we look forward to talking with you all soon.
Thank you, everyone.
Thank you.
Thank you.
Pleasure.
Thank you.
Thank you.
Thank you very much.